Welcome to the exciting world of cell biology. Here's a lab sample containing mostly protists, which are single-celled eukaryotic organisms. And what's really cool about them is they are relatively easy to view under a microscope. All we have to do is take our water sample, put a couple drops on a microscope slide, then apply a cover slip, and then we go searching for tiny organisms under our microscope. There are literally hundreds of independent living Paramecium in this slide, and we're just looking at a small area. Each time we zoom in, we can see more detail but see less Paramecium, which are just some of the protists in this sample. Now these friends are very active, so let's find some others that are less motile for us to view. This guy, well, the gender technically doesn't apply because Paramecium reproduce asexually and exchange genes through conjugation. Anyway, this specimen is less active, but is spinning so we can appreciate the 3D nature to it. One of the more obvious structures are the hundreds of beating cilia that surround it, which are kind of like tiny feet that allow it to move. Note that this is light microscopy, and light is penetrating through the Paramecium, and the colors are picked up through the various lenses of the microscope. All those lightly colored green and yellow to clear-like round objects, inside of it are organelles, or tiny organs. They're kind of like organs to us as humans are, and then complete various tasks to help keep them alive. Now let's find another specimen that isn't moving quite as much and see if we can make out some other structures. Now we got one that's cooperating with us. Now this isn't a living organism, and we don't want to harm our friends, so we're going to refrain from staining them in order to define the organelles better. But you can see the oral groove in the middle with the cytosome, that is the mouth. And then the Paramecium is going to consume its food through there, and then the food is going to be stored and transported through those green food vacuoles. We do see two prominent contractile vacuoles on the top and the bottom. The Paramecium has to regulate its water content due to osmosis, the movement of water. The water wants to move into the cell in a freshwater sample, so we have these two organelles to expel the excess water that it does so it doesn't rupture like an overinflated balloon. Now, if this wasn't cool enough, let's look at Paramecium under dark field conditions. Here is a specialized attachment. Notice the amount of light going through the actual slide. This is the standard light field setting, and here is the dark field setting. It blocks the light in the condenser and directs it obliquely, so we see the same Paramecium in a high contrast field of view with a dark background. Let's see what that same Paramecium cell looks like now. Now that's a cool view. The nucleus that contains the vital genetic information is actually a bit more apparent compared to our standard light field view. It is the darkened region towards the bottom of the screen. The contractile vacuoles are also a bit more distinct, as are the tiny food vacuoles and even the smaller vesicles. The Paramecium is oftentimes considered the white lab rats of cell biology because they're so widely used in labs and rather easy to work with, actually. While we're able to study this magnificent creature, one has to wonder, how aware is it of us? And it probably isn't much aware of us aside from the light passing through it. It follows our basic guidelines of what defines something as living, but its reality is that what it perceives, and it certainly has to be different from what we experience with our five senses. Thank you to our tiny friends that we normally can't see but are living all around us. Let's go explore the many wonders of how we operate at the cellular level.